everyone. So in this video, I'm going to speak about the lymphoid enhancer binding factor LIF1. I chose this protein because it has a very interesting but also intriguing behavior. So LIF1 is part of the high mobility group family and provides a paradigm because it has a very high affinity for specific DNA sequences, even though it has one HMG box, but is also poorly understood in many human tumor viruses. So now I'm going to start describing the LIF1 DNA interaction. See the NMR structure of the LIF1 HMG domain and the DNA, and I'm going to start speaking about hydrophobic interactions. So LIF1 protein encloses an extensive hydrophobic core. So here we can examine closely the labeled amino acids in green that complete the hydrophobic core, and we can also see here the ones in pink that extend this hydrophobic core, creating a dynamic interface between helices 1 and 2. So the HMG domain of LIF1 binds to DNA, creating hydrophobic contacts with AT base pairs that we can see here labeled in blue. So this domain is gonna wrap DNA around, inducing a highly distorted helix. So now we can see the different phases, like we can see how the DNA is being distorted, forming a wide and very shallow minor groove. Now I'm going to speak about some interesting cases, and in particular we have tyrosine 76. We can see here how it is inserted into the narrowed region of the minor groove, playing a key role in DNA binding. We can also see here how the aromatic ring of tyrosine is in close contact with the ribose ring of thymine and guanine. So polar side chains tend to contact the sugar phosphate backbone rather than hydrogen bonding to bases. I'm also going to speak about another interesting case, which is the case of methionine 11. So here we can see how its side chain is inserted into the base stack between these two adenine, disrupting base stacking but not base pairing. Opening of the bases leads to a large negative roll angle and an increase in rise. So here in AT to TA transversion result in a severe loss of binding affinity. In addition to the minor groove interactions, we can see that helix 3 terminate at proline 68. So this is going to induce a pronounced kink in the polypeptide chain and allows the C-terminal region to extend away from the core. So the tail of the protein is going to wrap around the groove. And as we can see here, it's going to position a sequence that is almost lysine and arginine residues. So their positive charge is going to interact with the high negative electrostatic potential and stabilize the distorted DNA